On this channel, we are passionate about enabling science in Africa. Today, we're going to be looking at microbiology, specifically which industries are impacted, then look at the extensive array of equipment that's needed to set up a lab. Additionally, we're going to look at the techniques that you need to implement for your workflows. And finally, we're going to contrast traditional micro with rapid methods. I hope you enjoy this interview and you learn something that's valuable to your implementation plan. Hi, today we're going to have a discussion on setting up microbiological labs in Africa. And I've got a guest on today, Ashika Harry, is going to give us an unpacking of, why do facilities ultimately need these micro labs and how does that benefit and protect consumers, patients, as well in the pharmaceutical industry. On that point, Ashika, could you give us a breakdown on, when it comes to setting up a microbiological lab, why do facilities and companies need this?
Hi, Jeff. Thank you for having me. I'm very excited to be here. So I just want to start off with a brief explanation of microbiology. So microbiology is basically the structure, the function, and the characteristics of microorganisms and how this may affect us in our daily lives. So the different industries that microbiology can be applied to can be the food and beverage industry. In this industry, microbiology is very important as it can detect for organisms that may cause spoilage of foods. So some examples are Listeria. As we know in South Africa, a few years ago, we had some of the Listeria outbreaks, and microbiology is important in this industry to restrict this from happening again. In the pharmaceutical industry, microbiology is important for the sterile production of drugs and drug products. The environment as well as the products used in the process should be monitored for microbiological contamination. In the cosmetics industry, we have the similar situation. Although cosmetics are non-sterile, they should be free of microorganisms. Similarly, in the cannabis industry, the cannabis plant as well as the materials used should be monitored for pathogens in sample preparation as well as in product. In the clinical industry, we have the susceptibility of microorganisms to antimicrobial agents. So as you can see, in all of these industries that the basic end goal is to look for microorganisms that may infect our end product and ultimately look at quality control procedures in this.
Awesome, excellent. I think that's a good rundown. And I often come across this because we do find a lot of companies that are looking to set up or expand into microbiology or offer those services or to have that as a function of the business. And it's always one of setting it up in Africa. What are some of the challenges customers come across in terms of it's sometimes remote, sometimes these suppliers are international companies, mostly based in North America. What do clients really need when it comes to setting up these micro labs?
Yes, Jeff, that is an important question as we see many challenges from most of our clients. So some of the common ones will be the lack of equipment and consumables locally. So as you just mentioned, that most of these companies are overseas, and to import them or even to worry about the servicing of instruments such as your laminar flows, flow hoods, your incubators, your autoclaves, that also becomes a problem. So often microbiology is misconceived as an expensive technology to implement. But this can be overcome if you look at partnering with a local distributor. One such distributor is Separations. So we are located in South Africa. Some of our importation channels are the USA, France, Germany, and the list goes on. Some of our partners that we work with on a daily basis include South Africa, Botswana, Nigeria, Kenya, and a lot of other African countries. So it's a service team. So they are factory-trained engineers that can assist you with your servicing, installation, as well as your calibrations and stuff for all of your equipment, especially equipment in the lab, such as your autoclaves, which need autoclaves or laminar flow hoods, which need servicing on an annual basis.
Okay, awesome, excellent. I believe there's also application support for people who need training in terms of trying to understand the workflows.
Yes, yes. So they are, and that is actually another challenge that is commonly seen, the lack of technical skills and training in some of the African countries. So Separations does have an applications team that's also service trained, and factory trained so that they can assist you with training programs and in any workflow that you may need.
Okay, awesome. Excellent. I mean, that's pretty comprehensive. And I think one of the nice things is like knowing that you need to do certain SANAS accreditation on pressure vessels for autoclaves, dealing with those issues ahead of them being a problem. That's something that it helps to have service and technical support for that. Now, I believe, Ashika, you'll be able to give us a better run through of the different techniques in microbiology. Can you perhaps share for us a presentation so we can kick into it?
So today I will just present an overview of the common lab equipment that you need in a microbiology lab, as well as the common methods and techniques used in microbiology. So this is a basic lab setup. You can see in the corner here that there's a laminar flow hood. There's also a water bath. Then you'll see other equipment such as analytical balancers and microscopes, which is commonly used in microbiology labs. So just before we go through the methods and the techniques, I would like to go through a list of equipment that's required in a microbiology lab. So for the sterilization of sterilization equipment in a microbiology lab, the most common one used would be an autoclave. So the autoclave uses moist heat sterilization, and this is commonly used for the sterilization of your cell culture media as well as other lab consumables. This can come in different configurations based on your application. They can be horizontal or vertical. Another method of sterilization could be dry heat sterilization, where you can use a hot air oven. So this typically sterilizes instruments such as pipettes or glassware. A Bunsen burner can also be used in the microbiology lab as a sterilization method. For this, you can vertically hold your item vertically into the flame, and this will sterilize your items. So this typically would be your inoculation loops, your spatulas, or even the mouths of bottles or test tubes.
Yeah, and I'll just mention something there, Ashika. I mean, this is the equipment that's often placed inside the laminar airflow that you mentioned earlier, which is, it basically gives you that assurity of sterility. If it's a properly calibrated and validated laminar airflow, your Bunsen burners inside that hood, along with the media that you migrate in. But I'll let you continue the equipment list because there's the general facility and there's also obviously the micro environment within the actual laminar airflow.
Yes, Jeff. So that is right. The Bunsen burner is situated inside the microbiological safety cabinet. The hot air oven and the autoclave is obviously not in the cabinet, but it's in the microbiology lab. For culture and identification in the microbiology lab, typical instruments you would require would be an analytical balance. So this is used to measure the mass of your granules or your powders or your liquids. Then the most important one would be your microbiological safety cabinet, which we just mentioned and we saw in the previous slide. So in a microbiology lab, you typically working with potentially hazardous or infectious microorganisms and also hazardous chemicals. And this could pose as a risk to the personnel that's handling this or also the environment. So what's needed is a safe environment to work in that can also be contamination free. So this is exactly the reason for using a microbiological safety cabinet, because it's used as a containment to work with your microbiological samples.
Yeah, and I'll jump in there quickly just because sometimes with a laminar airflow and a microbiological cabinet, there's a confusion. The one is if you're not dealing with anything hazard and it's just routine analysis, a laminar airflow is fine because it's just basically pushing air out, including to the operator. So if you're just worried about sterility and you're not worried about anything hazardous into that lab, that's where you use the laminar airflow. And exactly, when you're dealing with something that could pose a risk, that's where you have that microbiological safety cabinet.
Yes, Jeff, that's right. Then we can move on to a water bath. So a water bath is placed in a microbiology lab. It is a steel piece of equipment where you put in water in it, and it's regulated by a thermostat, which keeps the temperature at a certain required level. And this is commonly used for incubation or for thawing of your reagents used for media preparation. Then we can move on to our centrifuges. So centrifuges are used as a separation equipment based on density. So the centrifuge spins at a very high speed, and this creates a centrifugal force. So the centrifugal force pushes all your heavy material down to your chest tube or whatever you centrifuge in your centrifuge, and this will create a pellet. So this pellet can be used for further analysis in the microbiology lab. So centrifuges can come in also different configurations based on what you're looking for. Incubators are used as a temperature-controlled environment for the microorganisms to grow. It does have a glass door in the front, and it has a thermostat as well that can also regulate the temperature. Once the temperature is reached for the microorganisms to grow, that thermostat shuts off and the temperature is regulated in the incubator. Incubators can also come as shaking incubators, CO2 incubators, or even benchtop incubators if you're looking for that. pH meters is an instrument that's used to measure the alkalinity or the acidity of a solution in your lab. The refrigerators provides a low temperature environment for reagents or cultures that would otherwise not be stable at room temperature. So typically you would put in your cultures in your refrigerator, your prepared media, or even some reagents. This keeps them at a required temperature, typically at 2 to 8 degrees Celsius. Then we move on to the microscope. So microorganisms are very tiny and they're actually invisible to the naked eye. So this is where your microscope comes in. So a microscope is an optical instrument that uses a lens that magnifies even minute objects into a bigger visual object so we can see and analyze. The PCR thermocycler, so this is typically found in a molecular microbiology setting. It's actually used to amplify your DNA or RNA, and this is held at different temperatures for different steps of your amplification process, which are denaturation, annealing, or elongation or extension.
Yeah, so I think I'm going to jump in there. I mean, a lot of what you've discussed until this point with the PCR instrument is mostly traditional microorganisms, but traditional micro labs. In that context, one thing we do as well as separations is we often work in like a design suite where we can start placing equipment and give a visualization. Because one thing, whenever you're dealing with a new setup or building out a facility, you need to know what the footprint is on that equipment. You know, where does the laminar airflow go? Do you need more than one of them? You know, how many incubators do you need to position? If you need microscopes, what are you identifying? What are you characterizing? So that needs to be visualized along with the centrifuges or the autoclaves. And then additionally, like you mentioned, with the advent of rapid methods for testing, we're going to touch on that a little later in the presentation, but that's also becoming quite a hot topic in terms of finding ways to speed up the testing. So I'm glad you brought in the thermocycler here to kind of provide a bit of context for where traditional microbiology is starting to leave from where it used to be and start to be implemented for rapid methods. I'll let you continue.
Thanks, Jeff. Yes. So that is right. Some of these equipment is only needed in the traditional microbiology lab, but they can also be used in the molecular microbiology lab. You would still need an incubator. You would still need your water bath or a centrifuge for that matter, or even a microscope. So we can move into the basic techniques now of microbiology. So you'll see a flow diagram here. So the first one is aseptic techniques, and then we move on to culturing, incubation, and then enumeration, and then your identification of your pathogen. So in aseptic techniques, we need a sterile environment to work with so that there's no contamination. So this can be done in 3 different ways. Sterilization by using ultraviolet or gamma irradiation, or you can even use detergents for this purpose. Disinfection is to kill or inhibit any microorganism growth in the lab. Sanitization is the complete elimination of non-pathogenic as well as pathogenic microorganisms in the lab. Then we move on to culturing techniques. So before you actually place your microorganism in your culture media, you need to isolate or inoculate this. So these methods can be done by different methods. So this can be the streak plate method, the spread plate method, the pour plate method, or membrane filtration method. We'll go into this a little bit later in more detail, but for now I've just listed it here. So culture media is another consumable that you would require in your microbiology lab. So culture media is a source of nutrients for your microorganism to grow. So microorganisms, you have various different microorganisms and they all require different nutrients to grow. So therefore they have different types of media. So we can have your basal media, enrichment media, selective media, or even transport media. But we have various others as well. Culture media can come in dehydrated form, ready to use media, and we also have chromogenic media. So chromogenic media is a chromogenic substrate is included in this media, and when incubated with the pathogen of interest, this substrate will cleave and release a chromophore. So this leads to a color change in your petri dish, and this will help you with the identification of your microorganism.
Awesome. I want to jump in there quickly. I mean, often this is something that we come across often is that do I use dehydrated media? Do you use pre-poured? Do I use chromogenic? And I mean, the biggest advantage of dehydrated is the fact that you set the shelf life whenever you prepare it. So when it's in dehydrated form, you can stay on the shelf a lot longer. When it's pre-poured, you're kind of under the clock, you know. And it's also one of like, you know, if there was any issue with the manufacture of those pre-pours, you would notice immediately when you bring it out of maybe, you know, when you unpack it. But it's just something to think about as well. When you think about if you're really looking to do a lot of testing, dehydrated media often does prove to be quite economically viable, especially where labor costs are lower in South Africa than maybe we would see in Europe or North America, where you do see a lot of pre-poured methods. It really just depends on the volume, the capacity, and the use on site. So those are some of the considerations. And then chromogenic is extremely helpful for identification of certain things. But I'll let you continue.
Yes, and also, Jeff, chromogenic media does release the time to your result than traditional dehydrated media. So once your microorganism is on your culture media in your petri dish must be closed with the lid, and then this is sealed securely with tape or parafilm. And then we move on to the next step, which is incubation. So incubation, you obviously need an incubator for this. You place your petri dishes in here for a certain amount of time at a certain required temperature. This is usually at 37 degrees Celsius. Plates are usually inverted and stored in the incubator. This is basically to prevent any water droplets forming and falling into the media and thus causing contamination. Then we move on to the next step. So after your sample has been incubated for the maximum time that was allocated for this, you move on to enumeration techniques. So enumeration is an essential task in microbiology for counting the colonies that is present or has grown in your plate. So then we move on to the last step, which is identification of your microorganism. So the first step in identification is morphology. So this is basically looking with at your plate with the naked eye. So you look at the texture, the shape, and the size of the microorganism present. Then we also have staining techniques, where your microorganism can be stained with chemicals. And then this is looked under a microscope, and you look at the microorganism's characteristics. So other types of identification processes can be your biochemical tests. So this is based on the enzymatic activity of your microorganisms, or even serology tests.
Okay, awesome.
So now let's move on to those culturing techniques that I just mentioned in step 2. So I will just briefly go through each method, and we can look at how we can assist with that. So this first one will be the streak plate method. So as you can see, Jeff, we spoke about the Bunsen burner earlier. So this will be in the microbiological safety cabinet. And this is an inoculation loop. As you can see, the person is sterilizing this using the Bunsen burner. So once that's sterilized, the inoculation loop is actually dipped into your sample, which contains your microorganism. And then you have a Petri dish that has culture media in it, and you basically streak your inoculation loop around the corners of your Petri dish. So this is done. Once this is done, you close your Petri dish, and then this is incubated for the certain amount of time, and this can be viewed under the microscope for growth of microorganisms.
Cool. Or you can place that in the colony counter, if you need to. But you'll normally see the dilution factor that's applied across the streaking. Cool.
Yes, you can do that. So then we can move on to the spread plate method. So the spread plate method is a little bit similar to the streak plate. You have a Petri dish with culture media in it, but for this one, you're pipetting a certain amount of your microorganism sample, and then you're basically taking a spreader, more commonly a L-shaped spreader, and then you basically spread this evenly across the plate. Once this has dried completely on the media, then you close the plate, you seal the plate as per previous methods, and then you incubate this for the certain amount of time again, and then you take out your plate, and then you can view your colony growth.
Awesome.
So the next one would be the pour plate method. So this one is a bit different from the previous two methods. So this is actually taking an empty Petri dish, and you're pipetting your sample first into your Petri dish. Then you're pouring liquefied cool down media. So this is poured into your sample, and then this is allowed to solidify. So once this solidifies, then you seal your plate and you incubate it, and then you can look for growth of colonies. So this is typically used for looking at viable colonies and counts.
Yeah, 100%. And you mentioned the cool down media. Obviously, that media can't be too hot because if it is, it's going to obviously kill off, maybe potentially kill off microorganisms, which would impact the results. And I think at this point, yeah, perfect. I mean, we didn't mention quantification. Versus, like just confirming the presence versus quantification. I mean, and I think this might come up with sterility testing, when we specifically are looking for absence presence. I don't know if you want to maybe elaborate on that in terms of there are certain tests where you're counting, so maximum 100 colony forming units per centimeter, and then I think the other test is just absence presence. You want to make sure there's absolutely no microorganisms. So I think I've maybe explained it there for both of us and for the audience, but that's something to just consider as well, is are you quantifying or you're doing absence and presence testing? And that's also important in some of these regards, especially when it comes to viability of microorganisms, because sometimes, and this is some of the challenges with rapid methods, is you might pick up DNA, but if that organism was dead, then it doesn't speak to the viability aspect. So this is where the pour plate is quite a nice discussion, but I'll let you continue.
Yes, Jeff. So that will come obviously under your enumeration techniques. So there we can look at serial dilution methods or most probable number or even plate counts, as you just mentioned now.
Cool.
Then we can move on to the next technique, which is the membrane filtration technique. So this technique is usually used for your fluid samples and to look for microorganism contamination. So in this technique, you'll see the basic setup here. So this involves the use of a manifold, and this is connected to collecting flasks here, which is then connected to your vacuum pump, which serves as the pressure source. So here you can see just before the pump, there's a vent device here. So this is basically actually there to prevent any backflow into your pump, and thus protecting your pump and creating a longer life of the pump. So if we come back to the manifold, you'll see there's special adapters that fit onto the manifold. So these adapters are for your microfunnels. So we have different types of microfunnels as well. So this microfunnel is an autoclavable, reusable microfunnel. These ones will be your disposable single-use microfunnels. Sometimes these microfunnels can come with your membrane already attached to it. People in the pharmaceutical industry commonly use this, or even in the food and beverage industry, because of such high numbers that they actually testing for, or even the sterility issue.
Yeah, and I'll mention something else here because I mean that larger funnel you showed there. One thing that I do know these funnels have is they got a magnet, so that's actually operated with a single hand. So you just use one hand to declip it instead of having to struggle with a clamp and then reclamp that funnel. It's magnetic, so you raise it. You take your forcep, you place your membrane, and you just let it clip automatically in place. It's a really nice feature, especially if you're doing a lot of routine analysis. And the one thing I like about those disposable funnels is some of them even allow you to do sampling in the factory, especially in the pharmaceutical site. If you need to take water samples at site by the WFI tank, the water for injection, or you need to go just get samples throughout the facility, the ability to seal that, then move those samples to the microlab is definitely an added convenience, especially if you're constantly having to autoclave and sterilize shot bottles that are being used in that microlab. That actually adds up, having to constantly just, re-sterilize, and you don't know if those have been sterilized correctly. So, being able to use something that's pre-packed, sealed, do remote sampling, bring it to the lab, that makes a big difference. And I mean, I've seen even the migration away from manifolds to like these individual peristaltic pump formats where, it doesn't have this big waste trap and the vacuum pump. It actually does the same thing, but using a peristaltic format. So there's a lot of innovation we've seen over the years when it comes to the membrane filtration technique.
Yes, Jeff, you're 100% correct. So the magnetic filter funnel is magnetic. So I was actually coming to that when we have the membranes here as well. So these are different filter membranes that should be placed just before your magnetic filter funnel. And another feature of the disposable microfunnels is that some of them can actually be used as a petri dish as well.
Cool.
Yes. So, Jeff, another thing is that with the manifold setup, these are typically for larger sample volumes, and with the peristaltic format that you mentioned earlier, those are for lower volumes. It all depends on what the customer is looking for and your sample size.
Awesome.
So basically how this works is you put in your sample into your microfunnel, and then you switch on your pump. So your sample flows through your manifold and then collects into your collection flask. So once the sample has filtered through, you switch off the pump. The other feature of the microfunnels, they are made of plastic, so they're easily squeezable, and this can also be done with one-handed operation. So then after you take your membrane out of after your sample has filtered through, you put this on a petri dish which also has culture medium, and similar to the other techniques, you seal this nicely and then you incubate it for the required amount of time.
Yeah, so maybe just to unpack that. So the whole point here is to take a water sample that may or may not have any microorganisms, and then theoretically that membrane catches that, and then that's what enumerates on the actual petri dish. And in this regard, people often, I've seen this where they take shortcuts and they maybe say, oh, well, the filter membranes are maybe cheaper, sourcing them from somewhere else, versus a quality membrane. And the problem with that is the quality of the membrane is really important, because if you don't capture those microorganisms, you don't pick up that detection. And if you don't know if there is contamination in your facility, it could be after you've released multiple lots of a batch that you realize there was contamination. And just because you went cheaper on some of these membranes, you have to now return and restock and recall all that inventory because of that. And that's why those membranes are so important, because it really isn't in any way or sense, paying double the price even for a proper membrane is really important, because if you consider the costs of product recall, the reputational costs, you really want to just use the best membranes to make sure there is, in fact, any microorganisms caught early, you can do a corrective action and avoid all the major problems that develop. And I mean, let's be honest, if you're in QC or QA, quality control and quality assurance, having this type of test slip pass and not picking up detection early is definitely going to impact your ability to maintain your job, because that's the job of QC and QA, is to make sure they use the right materials to catch contamination early and introduce corrective actions. So I just wanted to mention that.
Yes, Jeff. And the membranes that we actually use from Paul have gridded lines. so it's easier for us to count those microorganisms that may be present. Also, they actually made of cellulose nitrate and cellulose acetate, so they're actually much stronger than a typical membrane. So all of these methods that we've just mentioned just now, they are traditional methods for traditional microbiology, and this does come with some challenges. So some of the challenges are listed here. So one, it is time consuming. So the time it takes to prepare this media, autoclave this media, and also autoclave all of your other instruments. It's also labor intensive. As we've seen, there are many manual steps, and this is all has to be done by the technician. So it's also very slow, so it takes several days for us to get a result. So this brings us to the rapid or the molecular microbiology techniques that you mentioned earlier, Jeff. So Separations is proud to announce our new partner, which we've added last year in 2021. So this is Biorad Food Science Division. So basically what we can offer from Food Science Division in Biorad is the molecular techniques around food, water, and environmental safety. So this technology is based on real-time PCR technology. Some of the products that we can offer include equipment such as the PCR thermal cycler, as well as automated liquid handling solutions for high throughput customers. We can also do consumables, which include the pathogen detection kits, and then obviously enrichment medias, as well as selective medias downstream. So this is the basic workflow of the PCR technology. The first step would be to enrich your sample. And then this is incubated for a certain amount of time based on which pathogen you are looking at. The second step would be your cell lysis and your DNA extraction. The good thing about the Biorad kits is that the extraction kit is included in the entire kit, so it's a one-box solution. So after this cell lysis and DNA extraction, and after incubating it for a certain amount of time, this is put into your PCR thermal cycler, and this is where your amplification happens and your detection, and you get results within 24 hours.
Awesome. So, I mean, this is just to give contrast. I mean, I know some of the traditional techniques, depending on the microorganism, can be not only days, but even potentially like 2 weeks to get enumeration and actually be able to complete that test. I mean, this is kind of game changer, especially if you consider inventory is an aspect of the business. You know, you're having to keep stock for release, and if you're waiting 2 weeks for a release test, it's quite a lot of inventory you need to hold on to and space in the warehouse. So I imagine that there's quite a strong adoption of these rapid methods, especially where it's allowed. Because, I mean, we didn't mention, for instance, sometimes when you talk about rapid methods in the pharmaceutical industry, that's far more challenging because there's the traditional, USPs that if you're doing the US pharmacopoeia or the EP standards, you can't deviate from the golden standard to some extent. But there's also the way to validate rapid methods. And, that normally has to be done for a while in concurrence with traditional methods before you can rely on that statistically. But I think it's quite a nice thing to see this adoption, and especially I think the food industry, maybe even in cosmetics, there's a big uptake of these rapid methods.
Most definitely, Jeff. As you mentioned, to hold a stock of food for such a long time and to wait for 2 weeks to get a result, that decreases your shelf life even further. And this is where the PCR technology is beneficial. Another thing about the validation, so the Biorad kits and the instruments are fully validated, AFNO validation. So they are a validated system and an alternative to the traditional methods.
Awesome. Great. Thanks, Ashika.
So just as you mentioned, Jeff, so this is a slide showing the differences in the time it takes for each and every method that we just spoke about earlier. So here you will see the standard reference methods. So with traditional microbiology, you will see it takes up to 7 days to get a result. With chromogenic or ELISA, which is an immunoassay, you will see that it does shorten the time to result, but it's still a long time as compared to PCR. So with PCR, you can see after enrichment, you get your result on the next day. So this dramatically decreases the time to result. So number one, the benefit is the speed. Number two, it's also very accurate as there's no, it is automated and there's no user actually reading the sample. Another thing is that because there is not much manual work, you have a lot of more time to do other stuff in the lab that you may need.
Yeah, so I get the sense that obviously the molecular techniques are far more precise because there's less user intervention. So not only in the culture, the enumeration, the counting, but a lot of these steps are removed, which does remove human error from this assessment and characterization. So no, that's a valid point in terms of like, and it frees people up to do actual constructive work in the lab. Sometimes the data analysis, the reporting, and not just manual techniques.
Yes, definitely, Jeff. The accuracy is one of the main benefits of the technology. So if we move on, we can see the different pathogenic kits that we have from Bio-Rad. So we have qualitative solutions as well as quantitative solutions, both for the food pathogens as well as water pathogens, such as Legionella.
Excellent.
Okay, so our next slide will show you the different separations workflows and how we can assist and which divisions are responsible for which industries. So you'll see the Biopharm Techniques department. They typically deal with the pharmaceutical, biotechnology, cosmetics, and cannabis industries. So for pharmaceutical, we can assist with sterile and non-sterile testing, environmental analysis, water analysis, or even cannabis testing. The biotechniques department will mainly focus on biotechnology in research and development and also the academic industry. The diagnostics department focuses on the clinical market. So this will be your sample processing, your diagnostic pathogen testing, antimicrobial sensitivity, which we spoke about earlier, and also environmental monitoring and identification. The water techniques department focuses mainly on the food and water quality control. So this is mainly your municipalities and your food industries. Our export department, so this is, they actually cover all of these different industries in sub-Saharan Africa. Please just reach out to us if you do need assistance with any of this. So our next slide focuses on the different key suppliers for microbiology from separations. So you can see Bio-Rad is our molecular solutions, where we offer the equipment such as the thermal cycler, the automated liquid handling, and then also the consumables such as the medias and the pathogen detection kits. From Paul, we can offer you all of our water filtration, membrane filtration techniques setup. So that includes your manifolds, your magnetic filters, your filter membranes, your microfunnels, and stuff like that. From Conda Lab, we can offer you all your dehydrated media, your chromogenic media, as well as your ready-to-use medias. So anything to do with culture media, we can get it for you from the supplier. From NUV, we can do all the lab equipment. So this includes your microbiological safety cabinets, your centrifuges, your autoclaves, incubators, and water baths. From Leica Microsystems, this is where we offer you our microscopes. From Soccerex, we can do pipettes and other liquid handling instruments. From Carl Roth, we can do all of the consumables such as petri dishes, inoculation loops, or your bacterial outspreaders. Alkanox is where we can supply you with the detergents required for sterilization or disinfection. And then Hirschman, we can do all of the glassware such as your beakers, your bottles, or your measuring cylinders.
Awesome. And I mean, this is the main suppliers. There are still some ancillary ones. If you're looking at automated workflows, there's quite a lot to be done with liquid handling robots. But this is a very good comprehensive list of the core suppliers that kind of go into this traditional micro as well as rapid or molecular techniques. Thanks.
Okay, so Jeff, that was actually my last slide, but I would just like to say that with microbiology, it's absolutely fundamental in all industries, and it actually affects us in our daily lives every single day. And on my last slide, we have Separation's contact details. So our head office is in Johannesburg, and we do have a Cape Town branch as well. So more recently, we have business development areas in the Eastern Cape and also in KZN. And then I also have my contact details if you do need to reach out to me.
Awesome, excellent. So thank you so much, Ashika. That was quite comprehensive. I hope it was educational for those looking for that. And for those who are looking for just an overview of some of the technologies, equipment, techniques to be applied in the lab, that was hopefully useful. And like Ashika and ourselves like to do is, I mean, we've got an applications team, we've got sales team, but everyone is quite involved in helping customers get set up and ultimately just enabling the quality of science in Africa. And that's a core focus of Separation. So Ashika, thank you for making the time. Really appreciate it. And I hope the audience found that useful. And be sure to go to the channel on YouTube for additional content because we like to produce these educational pieces to help enhance the overall quality of science. Thank you again.
Thank you, Joe.